One other model, which is a little bit different, and it's a takeoff of the conditional knockout of the NF1 alleles, and this is Jennifer's work, which she's going to be talking about at length today. But these mice, not only were they treated with the poly-I, poly-C to induce the CRE, but they were also given replication-competent retrovirus. And then the retrovirus would integrate into the genome at various areas, and that permitted these investigators to identify genes and pathways that potentially cooperate with NF1 to induce progression from MPD to AML. So upon infection with this replication-competent virus, these mice developed an aggressive acute myeloid leukemia. So they didn't just stay in the myeloproliferative disease, they actually progressed to an acute myeloid leukemia. And this is another animal model that has been used to try to understand better what cooperating genes may be working with NF1 in progressing from MPD to AML. And then there's some really very old data, but really good data from Chinquay. And this was some of the original SHP2 knockout mice, and these models were used more because this is pretty old data to understand what happens in the absence of SHP2. And if you just focus on this graph over here, this is data that was derived from murine embryonic stem cell-based differentiation, looking at differentiation in colony assays to primitive erythroid colonies, definitive erythroid, granulocyte macrophage, mixed, or mast cells, and at various times in culture. And what you can see, though, the key point here is that the black bars are from wild-type cells, and the hatched bars, which you can barely see because they're so low, are from the, quote, knockout cells. And this was really the first example that in the absence of SHP2, hematopoiesis is very dramatically reduced. And there was several more types of assays and studies done that demonstrated this point. And we also did some experiments a little bit later, and this was using the same mouse model except in a heterozygous state. And what we found in transplant assays was that the donor cells from the SHP2 heterozygous mice at varying concentrations compared to competitors resulted in lower chimerism. And so that suggested that there's also perhaps a hematopoietic stem cell defect in cells that lack SHP2. The downside or the limitation to all of these studies, even though there is a lot of work done with this animal model, was that there is a residual SHP2 protein product, and that's shown in this Western blot here. So for example, in wild-type cells, this is the wild-type SHP2, you can see in heterozygous cells, the mutant allele still produces its much lower level of expression. However, there is a residual mutant protein, and it's lacking amino acids 46 to 110. So, and then you can see in the homozygous cells, this protein, this deleted protein is there alone. So while this mouse was informative and yielded a lot of information, it's always a little bit limiting when you know there's this residual protein there and you're not really sure what it's doing. And so one of our talks today is going to be from Gordon Chan, and he's going to specifically talk about hematopoiesis and stem cell biology using SHP2 flox mice, so where conditionally the SHP2 allele can be completely deleted. And I don't know what the data is. I hope it's consistent with what was seen previously. But this should be a much cleaner model to give a better answer to some of those questions. And then obviously a key point now is to try to develop animal models bearing Sybil gain-of-function mutations. And there will be one talk today from Dr. Kenneth Liu, and he's going to talk about how bacterial, I think it's bacterial artificial chromosomes, I'm going to learn a lot from his talks, can also be used to generate basically knock-in mice. And these BACs provide advantages over just a traditional transgenic construct because there's much more regulatory sequences that come along with the gene. And also, he's going to talk about like a novel method of cloning, which is called recombinating. So we'll hear about that. So that's looking more into the future of potential future animal models that can be of use. And then Ben Braun will talk about the mutant KRAS-induced myeloproliferative disorder and treatment with a specific MEK inhibitor, PD-0325-901. And he'll share his findings with us about that. And then finally, Jennifer Lockley is going to talk about, she will use, again, the model of the conditional NF1 knockout then infected with the retroviruses, and look at various MEK inhibitors or PI3 kinase inhibitors to try to come up with understanding what some of those cooperating genes may be that promote MPD to the development of AML. So I think that's all I have.